Bam Bio.
Bam Bio! Bam Bio! Bam Bio!
Well, let's start with the basics. Everybody's got chromosomes. Humans have 23 pairs of chromosomes. A gene is a unit of heredity which codes for a specific type of trait. Different genes determine the different set of characteristics in an organism.
Now, an allele is a variation in a particular gene. This allows for people to have different eye colors, like me and my friend Brad's over here.
Everything in our gene makeup, or genome, is the exact same, except for one slot, which is different.
All chromosomes are happily paired together by size and by structure in a karyotype, which displays the complete set of chromosomes. Karyotypes can help determine whether you are a boy or a girl using chromosome number 23. If it's XX, you're a girl, and if it's XY, you're a boy. And with this lovely graphic right here, you can tell whether non-disjunction has occurred, where you could possibly see a sad loner single chromosome or an extra chromosome creeping on a happy couple.
What is non-disjunction, you ask? It's the failure of homologous chromosome pairs, or sister chromatids, to separate properly during cell division, resulting in an abnormal and unequal distribution of chromosomes in daughter cells. This isn't viable. However, if a gamete was to be fertilized, it can lead to the formation of syndromes, such as Down syndrome or trisomy 21, in which you have an extra chromosome 21.
Non-disjunction isn't the only thing that can cause problems within the body, because of random genetic mutations that can occur in the body that can change genetic material. The base sequence can be altered by either deleting or inserting other bases. Sorry, got fogged.
Very true, Taylor. Base sequence substitution can actually cause different amino acid coding, which can result in a different amino acid. This substitution can affect a protein and its function. A disease that happens due to this mutation is called sickle cell anemia, in which the hemoglobin located in our blood is an odd sickle shape, kind of like a C, and doesn't carry oxygen properly throughout our body. This is due to a mutation in which the GAG is substituted for a GTG.
All this talk about diseases is really bumming me out. Now let's talk about the types of traits that get you looking sexy. So basically, every cell in the body except for sex cells or gamete cells are diploid cells, which means that they have two sets of chromosomes, one from each parent. These dominant and recessive traits can be passed along for generations and can be displayed on a pedigree chart.
Some traits can be a combination of different genes working together, which is called polygenetic trait. Poly meaning many, and genetic having to do with genes. This can all get kind of confusing, but this just is called polygenetic inheritance, which has to do with the inheritance of polygenetic genes.
Biography, my favorite parts of the show. So if we're talking about genetics, there's no way we can bring it up without talking about Gregor Mendel, the father of genetics himself, also known as the P guy. Gregor Mendel was a friar born in July 22nd of 1822 in present-day Czechoslovakia. During his childhood, he spent all his free time beekeeping and gardening. And you'll never guess what he did when he became a friar. More gardening. This seemingly obsessive hobby may have damaged his social life, but he did a huge amount of good for us, resulting in a world-renowned pea plant experiment that we all know and love, in which he bred pea plants together to see what kinds of mixed matches it would take to grow certain traits, like flower color, yellow peas, green peas, wrinkly peas, brown peas. And after a crapload of experimentation, he established Mendel's law of individual assortment, which states that when gametes are formed, the separation of one pair of alleles between the daughter cells is independent of the separation of another pair of alleles which determine different characteristics which will be transmitted independently to the next generation.
Why do I care about independent assortment? What does it have to do with me?
Well, actually, Ryan, you uncultured buffoon, yes it does! It has a lot to do with you. Independent assortment occurs during metaphase 1 of meiosis, and meiosis is the reason why we have babies, and babies is the reason why we have you, so you better get down on your knees and thank individual assortment, you ding-dong!
Thanks, Taylor.
During metaphase 1 of meiosis, the mommy chromatids line up with the daddy chromatids, and each pair up starts entangling. And this is called crossover. And while those chromatids are smooching and groping, they start exchanging genetic traits at random, so they end up looking kind of mixed matched. In phase 1, they pull the newly shuffled chromosomes to the opposite poles of the cell, and once the gamete divides, it produces two totally different mixtures of that maternal and paternal data. And then those gametes proceed to create even more bodies of mixed up alleles. This results in different genes and characteristics in the offspring, and that is your biologaphy of the day.
Now that we've covered the basics on genetics, let's talk about how genetics can be used in our daily lives. Taylor, you want a piece of my orange?
Yeah, I'd love one.
No, that's genetically modified, right?
That's what?
Taylor. It's okay. As I've explained before, through our human genome, or our genetic material, we all have the same chromosomes between all living things. So we can insert and delete certain traits through biotechnology. Like in that orange right there. It's got no seeds, it tastes really good, it's nice and sweet, so enjoy it.
Well, then I will.
Like other GMOs, it can lower the cost of food production and extend sustainability. That means oranges for everybody! Yes!
And now, something completely relevant.
Genetic modification is the direct manipulation of an organism's genome using biotechnology. It is a process of changing the genetic information in existing cells, either through the insertion or removal of genetic material to modify a specific organism for the purpose of changing its natural characteristics. Genetic modification has many advantageous applications with the potential to be beneficial, possibly great promises and hopes for the future. For example, genetically modified organisms, or GMOs, are very prominent in agriculture and crop technology. Genetically modified crops can significantly increase and improve food production. Through genetic engineering techniques, these crops have insect resistance, fungal resistance, viral resistance, herbicide resistant, changed nutritional content, improved taste, and improved storage. Genetically modified organisms could reduce hunger since they are cheaper and have lower requirements to cultivate and grow. However, genetically modification is the subject of many controversies and concerns. There are also possibilities for harmful effects, dangers, and fears. The long-term effects of GMOs are not well understood. They could potentially be adverse to human health and the environment. Genetically modified organisms can be integrated into the wild if uncontrolled, and disrupt habitats and dominate over other species. A proliferation of genetically modified organisms could lead to a decrease in biodiversity. Also, the consequences of genes crossing species are not known.
Scientists have also started making clones of other organisms as well, not just oranges. Like Dolly the sheep, who is made from a group of artificial cells derived from one parent. At the same time, biotechnology is also widely contested because the long-term effects of GMOs aren't widely understood yet.
That's true, and that's why scientists continue to gain a deeper understanding of the genome as a whole.
And now for our closing act, Bam Bio's newest music video.
I'm bringing abundance back.
Yeah,
Those phenotypes just don't know how to act.
yeah,
Come over here so you can understand.
yeah,
Predict the genome that's along your strand.
yeah. Take it to the bridge.
Draw your squares. The parent gametes always come in pairs.
Uh-huh,
Align each column so they meet up there.
uh-huh,
So we can tell if they conceal or share.
uh-huh. Take it to the core. Go ahead, be dominant.
Learn to make.
Go ahead, be dominant.
Draw the grid.
Go ahead, be dominant.
Fill them all in.
Go ahead, be
Only
dominant.
partially dominant.
Go ahead, be dominant.
Heterozygous.
Go ahead, be
Homozygous
dominant.
could be either one.
Go ahead, be dominant.
Now we're done.
Go ahead, be dominant.
Put your punnet on.
Go ahead, be dominant.
Put your punnet on.
Go ahead, be dominant.
Put your punnet
Go
on.
ahead, be dominant.
Put your punnet on.
Go ahead, be dominant.
We hope you enjoyed today's episode of BAM Bio, and if you want to reference any of the information present, you can ask for a copy of the video, talk to Ms. Blaze about some private tutoring, or you can be able to review your textbook as well as SlideShare notes found on Moodle. Special thanks to John and Hank Green for the inspiration with their Crash Course web series. Please support the official releases on YouTube. Also, a special thank you to Dalton Grassel for his help as an editor and cameraman with the production.